from a lot of areas of biology, actually. I welcome students who are biochem majors or chemistry majors. It doesn't matter. You'll pick it up as you go along, and either one is fine. Probably heard of a virus. It's an infectious agent that makes copies of itself inside living cells. So you can see the little virus coming in, it infects the cell, makes a bunch of copies of itself, and those copies come out, and then they go on to infect other cells. So it's like a chain reaction. Viruses you've probably heard of, influenza virus, which infects cells of the respiratory tract, and of course, HIV, which infects cells of the immune system. Now, I don't work with any of these scary viruses that affect humans, so it's very safe. I work with something called a bacteriophage, or phage for short, which is a virus that infects bacteria. It doesn't care about humans, it just likes bacteria. The most famous bacterium in scientific research is E. coli, and E. coli comes from the intestines of warm-blooded animals, such as yourselves. The bacteriophage that I work with is called M13. It's a long stringy phage, and it likes to infect E. coli, make copies of itself, and then come out and then go on to infect other cells. Why the sewer cover? Well, M13 was originally found in the sewers of Munich, Germany, hence the M for Munich. Why was this bacteriophage in the sewers? Well, that's where E. coli is. Why is E. coli in the sewers? Because it comes from intestines. But don't worry, we don't work with sewage any more than we work with HIV, so no problem. All right, now all the information for an organism that leads to what that organism is like is in its genome, the DNA. And the information in your DNA, whether you're a human or a bacterium, it leads to the synthesis of proteins. And the proteins do most of the work in your body and give you all your characteristics. There they are. Your characteristics, your behavior, what you look like, what you do, everything. Now, if you have a mutation in your DNA, and this can be a tiny little mutation, then that can affect your proteins. Maybe a protein is made, but it's different. There's something wrong with it, or something better about it than the normal protein. Or the protein is the right protein, but the wrong amount is made, too much or too little. So that's going to affect your characteristics or your behavior, maybe sometimes for the better, but most of the time for the worse, so like a disease. Now, because we're working with this bacteriophage, it has a very small genome. It doesn't have a lot of DNA compared to a human, but to it, its DNA is very important. And if it has a mutation, and I'm showing a mutation right here, then that can lead to something different about the protein. The one we happen to be interested in is called protein 2, and we suspect, hence the question mark, we suspect that maybe more of that protein is made as a result of certain mutations that we know about. What we know for sure is it makes the phage go faster, meaning it propagates more quickly and makes more copies of itself in E. coli in a certain amount of time than it would if it didn't have the mutation. So we know the mutation leads to this. We don't know for sure the reason in between. So the type of experiment we do a lot is to actually quantify how fast the phage is propagating in the cells. So we take a flask, we put some medium in it, some cells, some E. coli cells, and some phage. A virion is just a phage particle, one phage particle. So we put in 1 times 10 to the 8. We let it go, so the phage is making more of itself in the cells, and then we plate, this is a petri dish, we plate and count the little blue dots, and that tells us about how much phage there was back in the flask. So at the very beginning, we aim to put, like I said, 1 times 10 to the 8 variants. Now, no matter what happens at the end, we always end up with about the same number of variants because there are only so many cells, and the cells get exhausted. So it doesn't really matter how fast the phage goes, we end up in the same place. But what's very different is in the middle, a normal phage with the normal DNA sequence will end up with about 5 times 10 to the 9 variants in this flask with 20 mils of medium. But if there's a mutation, we end up with a lot more. Now it looks like only 2 units on this scale, but it's actually a log scale, so that's 100-fold more. And that's a big difference. If you had a normal phage and a mutant phage in the same class competing for the same cells, the mutant would win. And at the end, you would only have mutants and no normal phage. All right, why do these mutations occur? Well, remember the phage from the sewers. That's this here, called M13. We actually work with an engineered version that a lot of molecular biologists work with called M13KE. And what the difference is, I'm going to rotate the one on the left so their genes are in the same place. So you see right down here, there's an extra piece that's not here. It was actually inserted right in this region here, and it's called LAC-Z. And that makes the size a lot bigger. It was 6.4 kilobases before, and now it's 7.2. So that big piece slows down M13KE compared to M13. So what happens with M13KE is it tends to pick up mutations. Not that they undo this big insert, although they are nearby, but they're not undoing the presence of that insert. That insert is still there, but they compensate. They do something that compensates for the slowness that came from that big insert. So it's kind of like evolution, survival of the fittest. Any phage that picks up the mutation now has an advantage of one that didn't. And this graph just shows that that's the normal wild type M13 from the sewers. Turning it into M13KE made it slower. Picking up a mutation makes it faster again. How do the mutations work? And this is the part we're working on in the next couple of years. We'll really be working on this a lot. We've discovered many, many different mutations, and they're all in this little area in front of gene 2. So we think there's something to that. There's something that those mutations are doing to the amount of protein 2 that is made. And protein 2 is important in the life cycle of the phage. When the phage is making copies of itself inside the cells, protein 2 is very intimately involved in that. So if there's more protein 2, you expect that that might go faster, and hence the faster propagation rate. So we have to figure this out. We tried a few different ways, and we're still working on proving that that's actually the case. So what do we actually do in the lab? And here are some of my lovely research students. Because we work with phage cells, we have to be sterile. We have to have things autoclaved. So here we are getting ready to go to the autoclave. This is Marta, Emily who's in the room, and Lauren who's in the room. And here's Emily putting some cells onto a plate. Some of our E. coli. Organization is very important in my lab. We deal with hundreds of little microcentrifuge tubes, and they have what looks like water in them, but it's not water. It's all our phage and our cells and DNA and all kinds of things. We have to keep it all very organized. Labeling is very important. So if you like being organized and dealing with lots of little things, then hey, this is the lab for you. So yeah. And then lab notebooks, big deal for us. Our lab notebooks have to have all the information that tells us exactly what every single one of these tubes is. And we'll go back to tubes from 8 years ago, and we'll look back in someone's lab notebook to find out where that is. Yeah. And last slide. And plating phage, here we go, putting phage on the plate. And here's Marta showing the plates. We count the little blue dots that tells us how much phage we have. And thanks to people and places, and all my students over the years, and we collaborate with someone at New England Biolabs, which is in Ipswich, and we visit this lovely